All right, so again, if you ever have a question about why I graded something the way I did, feel free to come to my office. A couple of you did, so that's awesome because that will help you make sure that you understood the concepts really well. But if you don't want to, that's fine too. It's up to you. But I am in my office for that purpose. So during office hours, feel free to come by and clarify anything that you're not sure of. So in this chapter, chapter 4, we're going to start jumping into some of the details of cellular anatomy. So of course, first we're going to talk about the prokaryotic cells, and then we'll talk about the eukaryotes. So again, I assume that a lot of this is hopefully review from other classes you've taken. The idea that, you know, there's organisms that don't have a nucleus and some that do, and that's a pretty major difference. So if it's been a while since you've covered that, that's okay. It's been a while since I took intro bio too, so I totally get it. But, you know, those are going to be some really fundamental concepts. So they're going to be like classic fundamental concepts in microbiology that I will ask you about again and again and again. And you'll be like, oh my God, she's asking about this again. And people will still miss it on the exams. Okay? Like, you'll be like, stop, it's the 4th time you said it. I know, it's the 4th time I've tested you on it too, and still. Okay? So these are absolute fundamental differences that you just have to memorize, and you just have to know them. Okay? So of course, the major difference is that without exception, really, a prokaryote does not have a nucleus. The genetic material is not surrounded by that membranous structure. If you get into more advanced microbiology classes, you'll find that there are some microbes that have evolved this sort of protonucleus, but not a nucleus like we see in our eukaryotic cells. Now, again, I really want to highlight this word typically, because sometimes people get hung up and everything is absolute, right? So nothing is absolute. Prokaryotes typically have a single chromosome, but I can think of at least a couple examples off the top of my head where there are more. It is typically circular, but I can think of examples off the top of my head where they are linear. Now, in order for that DNA to be packaged within the cell, remember, prokaryote doesn't have a nucleus, so we're not packaging in a nucleus, but we still do have to package it in the cell. It is typically coiled on itself, like super coiled, like really, really, really tightly wound. And it free floats in a region of the cytoplasm that we call the nucleoid, right? It's not a nucleus because it's not membrane bound, but it is the region of the cell where we find the DNA. Now we can contrast that with the eukaryotes. Eukaryotes typically are diploid, meaning we have pairs of chromosomes. They are typically linear, but some organisms are weird. There are some eukaryotes that actually exist in most of their life as haploid, and then every once in a while we'll flip to diploid and be like, hey. Right? And then you've got plants which are really weird. They're like, I'm octoploid. Like, that's fine too. Do whatever you gotta do. Now, eukaryotes have an extra challenge when it comes to packaging their DNA, because not only does all of their DNA have to fit within the cell, it actually has to fit within the nucleus. So eukaryotes have constrained their genetic material to this organelle. In order to do that, eukaryotes also wind their DNA, but eukaryotes wind their DNA around protein complexes that are called what's a nucleosome when you have the protein and the nucleic acid, and that's made of proteins called histones. So DNA, and it's wrapped in a very specific way. It's not haphazard. It's like a specific number of nucleotides wrap around the histone. There's a specific gap of nucleotides and then another winding. Okay. Prokaryotes do not have membrane-bound organelles. And I'm going to tell you right now, and I don't keep secrets. I will literally ask this question probably on 2 different exams that involves something about where organisms do the electron transport chain. More than 50% of you will say prokaryotes do it in the mitochondrion. I'm telling you right now, and you're all laughing, but literally this is the 4th time I've taught this class, and I have this conversation every time. Prokaryotes don't have a mitochondrion. They can't possibly do the electron transport chain in the mitochondrion. They don't have one. They do it in their plasma membrane. So if you haven't noticed yet, I tend to give a lot of spoilers if you just listen to the lecture. So if you're here and you're just like goofing off on your computer and you want to get the points for whenever we're here, that's totally fine, but you're going to miss like 50% of what I'm going to ask you on the tests. Because I know you can read the book, I know you can read the slides, but you come to class because, in theory, you want to learn from the instructor. So the instructor is telling you, prokaryotes don't have mitochondria. They do the electron transport chain. They do not do it in the mitochondria. They don't have Golgi. They don't have ER. They got nothing. They're actually relatively simple. Eukaryotes, on the other hand, have many membrane-bound organelles. They do the electron transport chain in the mitochondria, the powerhouse of the eukaryotic cell. They may have chloroplasts that allow them to do photosynthesis. They have Golgi. They have ER. They have all these vacuoles. They have a lot of stuff. Prokaryotes typically have a cell wall. Not universally. Not every prokaryote has a cell wall. When a prokaryote has a cell wall, members of the bacterial domain have a cell wall made of a compound called Peptidoglycan. And that is universal. If bacteria have a cell wall, it is always made of Peptidoglycan. We have never found a bacteria that has a cell wall made of something different. There are bacteria without cell walls. They always have Peptidoglycan if they have a cell wall. The same thing with archaea. There are some archaea that do not have a cell wall. You don't have to have one. But if they do, it is always made of pseudomurin. We have never found an archaea with a cell wall that is not pseudomurin. Some eukaryotes have a cell wall. We don't. That's why we're squishy. Plants do. That's why they're not squishy. And their cell wall is going to be made of some sort of polysaccharide. Things like fungi can have cell walls made of chitin or glucan or mannan. Plants tend to have a cell wall made of cellulose. They're all polysaccharides, just different polysaccharides. Prokaryotes typically divide by binary fission. Not always. There are some that do different things, but typically binary fission. Eukaryotes by mitosis. Remember, prokaryotes can't do mitosis because we don't have a nucleus. The nucleus, right? Mitosis implies the nucleus dividing. So again, these are these kind of big picture differences between prokaryotes and eukaryotes that, you know, if we like required an exam of you at the end of getting your biology degree, like you needed to complete a terminal exam to get your biology degree, that kind of question would be on there. That's how fundamental it is. Every single person with a biology degree should know the difference between a prokaryote and a eukaryote and be able to articulate that. It's not even just for micro, it's for all biology. That's just a classic biological truth. So prokaryotes, again, typically, are unicellular organisms. Sometimes they'll form like associations with one another, but they don't have like the differentiation of tissues like we see with some of our multicellular eukaryotes. So they're almost always going to be, you know, the lone organism, the lone cell. They may be in a community, but each individual cell is its own full organism. Again, they don't have a nucleus or other membrane-bound organelles. And the term prokaryote includes both bacteria and archaea, though we classically focus on bacteria in a class like this because they're far more common. So bacteria, they are prokaryotes. They vary in size and shape. On average, again, right, we don't like to deal in absolutes. So you can't say that a prokaryote is always smaller than a eukaryote. That is not true. There are prokaryotes that are far larger than some of our eukaryotes. The unicellular ones. I mean, you're not going to see like a prokaryote that's this big. But some of them can be much larger than our unicellular eukaryotes. So, for example, this bacterium here is 3/4 of a millimeter in diameter. You can actually see that with your naked eye. But the smallest can be very small. For example, Mycoplasma genitalium. This is our smallest free living. It's not like an endosymbiont. 0.2 microns, very, very tiny. So again, typically, yes, they are smaller than eukaryotic cells, but you can't say it as an always. So if I asked you what are some differences between prokaryotes and eukaryotes, and you say prokaryotes are smaller, I'm not going to give you full credit for that. If you say prokaryotes are mostly smaller, that is true. So morphology. Bacteria and archaea can have different shapes. Their cells can have different shapes. Most of the time, if a bacterium has a shape, it is always that shape. Like Staphylococcus is always a coccus. It's never going to be a bacillus. It's always a coccus. There are some bacteria that are what we call pleomorphic. And they're not going to be so dramatic that, a coccus changes into a spirochete. But it can be a bacillus can shorten and look a little bit more like a coccus and then start to elongate again. That's typically what we see with pleomorphic. You might see some that are a little bit longer than others of the same species. And we also can talk about how the cells arrange themselves in three-dimensional shape. So our three classic shapes in bacteria are, of course, the coccus, which is round, the bacillus, which is, we call it rod-shaped or elongated, and then spiral. Spiral can sometimes be as simple as like a comma shape, like Vibrio, or it could be more like a corkscrew, like some of the spirochetes, like Borrelia or Treponema. So these are our kind of like three classic shapes of our prokaryotes. And again, typically if you're talking about a pleomorphic, you'll sometimes see like the bacteria that causes diphtheria can be pleomorphic. So the rods can be kind of long or they can be kind of short. But a rod, just in general, could also be a longer rod versus a shorter rod depending on the species. And I know that even looking under your microscopes at like 1000x magnification with the oil immersion and everything, sometimes it's hard to discern like the rod shape. Except for Bacillus megaterum, that one's like massive. That one's easy to pick out. There are some more unusual shapes if we move over to the archaea. For example, there's this genus that has a kind of a star shape. There are some that are just like solid rectangles. So again, we can see different shapes depending on which type of organism they are. But we typically wouldn't see a coccus go all the way to a bacillus to a spirochete. We don't see that type of change. And we can also talk about arrangement. And sometimes, like in medical microbiology, people will ask questions like, well, we know that Staphylococcus grows, right, like this, in these grape-like clusters. So why, if I'm looking at a microscope and I've done a good gram stain, you trust your gram stain. I've done a good gram stain, I know that this is a gram-positive, coccus-shaped organism and I see it in clusters. Why can I not definitively say that this is Staphylococcus under the microscope? Right, it's a three-dimensional arrangement. The way you make the smear, the age of the culture, the amount of culture, all of that can change. So the arrangement under the microscope could give you a hint. It might give you a starting point. I see this gram-positive coccus. It looks like it's growing in clusters. I'm going to start looking at staph, but don't get obsessed with the idea. It could be something else. But typically that's what we would see. So you can have single organisms, single coccus, diplococcus. This one is actually a pretty reliable indicator when they grow in those pairs. So in a clinical setting, gram-negative diplococcus, very informative, depending on where you got the sample from. You're almost certainly looking at the genus Neisseria. And if you get it from the cerebrospinal fluid, you've got a patient with meningococcal meningitis, Neisseria meningititis. If you get it other places, you have Neisseria gonorrhoeae. Gram-negative diplococcus, very uncommon in a clinical setting, so that can be very informative. If you have long chains of coccus, we call that streptococci. Sometimes that implicates the genus Streptococcus. Maybe your patient has strep throat or group B strep. You never know. They can grow in tetrads. I don't know why they don't call this like an octet, but that's kind of what it is. A group of 8, and then of course the clusters. We have fewer arrangements that are possible for our Bacillus species. You can have a single, a diplo, a chain, or you have this Coccus Bacillus where they're not a coccus because they're not totally round, but they're also not very long either in terms of a rod-shaped bacterium, so we call that Coccus Bacillus. And then the spiral bacteria are real easy. They don't have any arrangement. They always hang out by themselves. So I told you the spiral bacteria, it could be as simple as this curve, right? This comma shape, that's Vibrio. You can have kind of more curves, and that could be Spirillum. Then when you have these really, really thin bacteria that look a lot like a little corkscrew, those are almost always spirochetes. So they have a helical shape, and here the Spirillum have a rigid body, and then they call these helical shape with a more flexible body. And especially the spirochetes are usually really motile because they spin just like a corkscrew. Makes it really easy for them to burrow into tissues. And again, they typically there'll be a community of them, but they don't have an arrangement where they're like attached to one another. So questions about morphology or arrangement before we get into some of the anatomy and parts of the cell. So again, as you're in the lab and practicing, making your own stains, looking at prepared slides, just making your observations under the microscope, see if you can tease out these things. Like, I know this culture is supposed to be Staphylococcus. Am I seeing those clusters? This one is supposed to be, Streptococcus. Am I seeing that? So it's always good to practice making those observations. So broadly then, when we talk about the anatomy of a prokaryotic cell, these are the pieces. So they've differentiated in this image. You have some features that are in red and some features that are in black. So red, all of the features that are indicated with a red font, that represents things that every bacterial cell has. We have not identified any exceptions to the red. They gotta have the red. Black, those are your bonus features. Maybe most bacteria do have them. Most bacteria do have a cell wall, but not all. So that's why it's not red. So red is mandatory, black is optional. And so the way that we'll start talking about prokaryotic cells and their anatomy is we'll start as far on the outside as we can get and move our way in. So if we want to start as far out as we can get, we talk about structures that are technically outside of the cell wall or if we're talking about gram negatives, which we'll get to in a little bit, we talk about things outside of the outer membrane, outside of the cell. So outside of the cell, some cells can make something called a glycocalyx. It's a sticky, mostly polysaccharide, sometimes loosely, sometimes more tightly organized structure. So it's a sugar coat. So it's viscous. So that means like it's not solid, but it's also not a liquid. It's jello-y. Mucusy is a really good way to describe it. Kind of like the texture of mucus. It's made usually of polysaccharides. Sometimes there can be proteins in there as well, but it's mostly polysaccharide. And again, there are a couple of different ways that bacteria can make a glycocalyx if they do. Some of them make a capsule. So a capsule, kind of like last week on Thursday when we talked about different kinds of staining and we talked about capsule staining and how a capsule is this tight structure associated with the cell. So it's a more tightly organized, it's attached to the cell wall, but it is this polysaccharide, maybe polypeptide mixture. The slime layer is a little bit more loosely organized and loosely attached, but often they can serve very similar functions. And typically a glycocalyx, at least in the context of infection, because I know we often think about microbes from that perspective of what do they have that can make us sick. So that's looking at it from that lens. This glycocalyx, regardless of whether or not it's a capsule or a slime layer, can contribute to virulence, that ability to cause disease, through some different mechanisms. One, it's sticky. That can allow the bacteria to adhere. It's actually really hard to stick to us. We have a lot of mechanisms to prevent that. Like we have our respiratory secretions. We're always like getting rid of mucus. We urinate. We blow our noses, right? We breathe in and out. We have those nose hairs that get in the way. We try to do a lot to protect ourselves. So sticking to us gives them an advantage because they can stick. Can't cause an infection if you're not even there. So at least you're there. The other thing that this polysaccharide glycocalyx can do is inhibit phagocytosis. That's one of the major mechanisms that our immune system has, our innate immune system, to recognize and destroy pathogens. They are not, our immune cells are not very good at recognizing and destroying bacteria that have a glycocalyx or a capsule. So if you've taken genetics, you probably talked about that classic experiment with the rough and the smooth bacteria and how we figured out that bacteria can exchange genetic material. What we see is that bacteria, same species of bacteria, one strain can make a capsule, one strain cannot make a capsule. The one that can't make a capsule, no big deal. The one that can, kills. The only difference, the capsule. So that ability to stick and evade the immune system is really, really critical. But before these organisms evolved to infect the available hosts, making a glycocalyx also has important benefits for environmental bacteria. Bacteria that don't cause infection at all still can make a glycocalyx because it allows them to form a biofilm, this community structure that can allow them to perhaps stick to rocks in a river or whatever it is they want to stick to. But it also can prevent desiccation. It's gooey. It's mucusy. There's moisture in there, so it can prevent them from drying out. And again, it's a big mixture, mostly of carbohydrates, maybe some polypeptides. You can eat it too. So it can provide a nutrient reserve. So it has a lot of benefits for bacteria that cause infection, but it also has a lot of benefits just for the bacterium itself. So again, why would they have evolved something like this prior to having organisms to infect? because it prevents them from drying out and it serves as a nutrient reserve, allows them to stick into their environment. Now, in terms of infection, how many of you are pre-dent? Anyone want to go to dental school? A couple of you? Yeah. This is one reason I would never consider going to dental school. So bacteria in our mouths, right, are able to stick using a glycocalyx to the surface of our teeth and form a biofilm. That's your dental plaque. And of course, we're all very good. We remove that plaque twice a day by brushing our teeth, and we go to the dentist and get our teeth cleaned, right? All of that. If not, right? So eventually, right, those biofilms can cause inflammation of the gums. They can get down into the roots. They can cause periodontal disease. You lose your teeth. It's a big deal. So again, that biofilm is really great for them, not so much for us. So Streptococcus mutans is a bacterium that is capable of doing this. Very, very common in the oral cavity. This is why they recommend if you have kids, like you don't kiss them on the mouth, no matter how cute your babies are, don't give them a little kiss, because you have that bacteria, they're not born with it. That's how they get it. One of the most common bacteria that causes caries or cavities. Questions about the Glycocalyx, its role in virulence, and just its role for bacteria in general. Okay, so we're still on the outside. Some bacteria are motile, they can move. And of course, bacteria don't have arms and legs. They most often move using a structure called a flagellum. So the flagellum, for those bacteria that have it, is very important. We tend to see that if a bacterium is supposed to have a flagellum and we are able to prevent it from making the flagellum, it can't cause infection, or it causes an infection that's really easy to clear. So for those bacteria that make a flagellum, it's often very, very important. We call it the H antigen. So there are versions of the flagellum. So when we talk about, for example, you've probably heard of E. coli O157H7. That's the one that, is often found in raw beef or undercooked beef that can, they call it hemolytic uremic syndrome, so it can get out of the GI tract and destroy your bladder and kidneys. It has that H antigen, the flagellum. There are three major parts, so I guess this is why we talk about the glycocalyx first, because there is a part of the flagellum that will be embedded in the cell. So that's how it attaches. So they have a filament. It's made of repeating units of a protein called flagellin. It has a hook right here, and then it has this rod ring structure, and you can have them in gram positives, which is shown right here, or in gram negatives. So both kinds of bacteria can make a flagellum, right? But not every species does. Not every species is motile. But the ones that are, those flagella tend to be very, very important. They can change the expression of it. They can increase the expression. Like if they need to be motile, perhaps the environment has changed and they want to leave, they can increase the expression and make more flagella. They can also, if they are like, no, this environment's pretty cool, let's sit down and make a biofilm and hang out, they can downregulate the biofilm, or the flagellum. So they can turn its expression on and off. But again, if one makes it and you take it away, that usually inhibits its ability to cause infection. So not only do we have this structure, the flagellum, they can also be found in different arrangements. And again, what we tend to see is if a bacterial species has a particular arrangement of flagella, that is its arrangement. So it doesn't change flagellar arrangements. So we have peritricious, which is where you have flagella that can cover the whole surface of the organism. You have monotricious, where they have just one, and it's usually polar. Oh, one thing is Coccus bacteria don't have flagella. Because they're round, they would just kind of spin in place. They have to have some sort of polarity, so they usually will be rod-shaped or sometimes comma or spiral shape. But yeah, if you put a little rotor on a ball. It's just going to roll. So you're not really going to have much going on there. Okay, so you can have monotricious and polar, where it's at one end. You can have lophotricious, where you have a clump of flagella. This is polar at one end. You can have amphitricious, where they're at both ends, or both poles. So I believe you have a slide that looks very similar to this in the lab. It's a sporillum. You also have a really nice peritricious one. It doesn't look like this because it's not an electron micrograph of Proteus, I believe Proteus vulgaris. And so how does that help them move? So when bacteria move, we call it taxis. I know it's spelled taxis, but we pronounce it taxis. So motility is taxis. And microbes can move either to an attractant or away from a repellent. And those attractants or repellents could be things like various chemicals, maybe nutrients or toxins. It could be light. Maybe they're attracted to light. There are other bacteria that are actually, afraid, right? I mean, it's a bacteria, it's not afraid, but they move away from the light, so they say they're afraid of the light. They could be moving to oxygen or away from oxygen. They can move in response to magnetic fields. There are a lot of factors that can cause bacterial motility. But the challenge is that bacteria don't have a nervous system. It's not a directed movement, and it's not really a decision. I smell coffee in the morning. That is a decision that I make to go have coffee. I do that for your benefit. I love coffee. I mean, if I smell a pot of coffee, I'm moving in that direction. But bacteria don't make that decision, and maybe I don't make that decision either because it's like 5 in the morning. I just sort of stumble downstairs and go have coffee. So then how do bacteria know that they should be moving toward something they need or away from something that's bad for them? It's all chemical signaling in the cell. And often what that chemical signaling will control is if it is something that requires them to move, they will rotate the flagella in a particular direction. So in this example, they're showing a bacterium with peritricious flagella. So you know it's peritricious because they're all over the cell. So this bacterium with peritricious flagella, because of the chemical signaling, coordinates the rotation of all of the flagella, and they all rotate in a counterclockwise direction. When that happens, the bacteria enter what's called a run motility. So they just kind of move in a straight line. And again, they can't smell, they can't see, they don't know. They go based on the chemical signaling, usually based on the concentration of whatever chemical or the intensity of the light, or the available amount of oxygen, whatever that signal is. So after a little bit of running, at least in the case of these bacteria, the bacteria will then tumble. And in this case, tumbling involves changing the direction. So instead of the rotation being counterclockwise, the rotation will go clockwise. And the bacteria will determine the concentration. The concentration is higher, then they start running again. The concentration is lower, they'll tumble, right? So they go through this run and tumble. They eventually will make it to where they need to be, but it's not that direct path. So if maybe you're not a morning person, but you set your coffee and it's kind of dark and you're kind of not awake and you like bump into the wall here and you bump into the wall there, but eventually you make it to your coffee. That's kind of how it is. You got turned around because you stepped on the cat. You can tell what my house is like in the morning. You stepped on the cat, you stepped on the dog, you ran into a kid, I don't know. Eventually I make it to the coffee. Eventually the bacteria get to where they need to go. Do all bacteria move that same way? No. Another really classic example is Pseudomonas. It has one flagellum. If it moves its flagella, I believe counterclockwise, it goes forward. If it moves it clockwise, it goes in reverse. So it can only move like forward and backward. It doesn't do like the run and tumble. So it relies on that Brownian motion, again, why we make you take physics, just kind of that things move to get reoriented so it can start going again. And we can determine motility in a number of different ways in the lab. You could always do a wet mount, right? Especially like with that phase contrast or dark field microscopy, you can actually see the bacteria moving, like the individual cells. Classically, what you will do, some of your biochemical tests that you'll perform later in the semester involve what we call a semi-solid agar. It's not quite as thick as your classic agar. It's a little bit less gelatinous, and that allows bacteria that are capable of doing this movement to spread out. So you can actually see how the bacteria have spread out away from the initial site of inoculation. So again, motility is a really important feature for those bacteria that have it. Others just rely on, well, wherever the wind blows, or the water moves, or the blood, or whatever. Wherever you got, that's what you get. And then there are the spirochetes, which are always very weird. So spirochetes are weird. They also have flagella, which is not weird, because we just talked about bacteria that have flagella. They put theirs on the inside. And so you might go, okay, you put your flagella on the inside, how do you move? That's like, putting your boat, like if you have a boat, I don't have a boat, obviously I don't have a boat. If I had a boat and it had one of those little, propellers and I put it inside the boat, how is that gonna help me go boating? I'm gonna be stuck there with my boat motor on the inside and not doing anything. So how do bacteria move if they keep their flagella on the inside? Well, they have this really cool evolutionary adaptation where they basically wrap it around their cell wall. And they call them either endoflagella because they're on the inside, or they call them axial filaments. I think that word is boring. I like endoflagella better. And the flagella can still kind of move, but it's different. That's what gives them that rotational or corkscrew motility. Again, this is what helps them to penetrate tissues. And you can see this really well when you look at spirochete infections. You can see how they have moved through the tissue. Right? Treponema pallidum is what causes syphilis. And what you can see is where the bacteria were able to burrow in the skin, they start to move and they cause a wound, a lesion, as they move through the skin, literally rotating through the skin. Borrelia burgdorferi causes Lyme disease. Tick bites you, the bacteria spread, and you can see the redness in the skin as the bacteria spread. So that ability to be a corkscrew, they don't have to have any special enzymes. Most of them don't. They don't have any special enzymes to allow them to degrade connective tissue or to break apart cells. They are a corkscrew. They literally just burrow in and go where they want to go. So that evolutionary mechanism of keeping those flagella on the inside, one, protects them from being recognized by the immune system, and two, allows them to have reduced their genome. They don't have to make any fancy enzymes, and they can still do the same thing, burrow in and cause an infection. Because the flagellum is a big trigger to the immune system. We don't make a flagellum, not in the same way. So when the immune system sees that, it's like, ooh, this is bad. We should try to attack this thing. So let's hide our flagella. Now we have nothing to attack. Questions about flagella or motility or taxis? Cool. All right. Okay, we're still outside. We'll be going inside in just a minute. So the last kind of external to the cell wall structures, and again, these are all bonus features, none of these are mandatory, are Fimbriae and Pili, and these tend to only be seen in our gram negatives. They have different functions. So Fimbriae are going to be these long hair-like projections. They're not flagella, right? Because they're made of different proteins. They don't have that rotational motion. They don't really help with motility. But they do help a lot with attachment. And again, if you have two members of the same species, let's say you have two Neisseria gonorrhoeae or you have two E. coli, one can make Fimbriae and the other one can't. The one that can is far more likely to be able to cause infection. These bacteria can sometimes also make a structure called a pilus. Multiple would be Pili. Usually there's only one or two per cell. These can also sometimes help with motility. But some bacteria that make a pilus do one for one very important purpose, and that's for genetic exchange. And we'll talk more about that, I think, in chapter 8. But some bacteria can make a pilus that allows them to move DNA from one cell to the other, so they can share genetic material with one another. Okay. Are we ready to go to the cell wall? Okay, let's do it. Cell wall. Again, not every bacterium has a cell wall, most do. The cell wall generally functions, 1, to help maintain cell shape, 2, to help protect the cell from changes to the environment. It can act as a little bit of a barrier. It is not solid, so don't picture like a brick wall. It's a little bit more than a chain link fence, but like a wooden fence, right? It's pretty solid, a wooden fence, but there's also gaps. So the wooden fence helps you a lot. Things can come in and out, but it protects you still too. And it can also help protect against osmotic lysis. It can protect the cell from bursting. Because again, it's rigid. Eventually it can, but it can hold together for a while. So those bacteria that don't have a cell wall, they tend to have more things like cholesterol and really tightly packed membranes to help protect against osmotic lysis. And they tend to be round, because if you are a cell, you're going to be round unless you have a wall telling you to be another shape. Because most bacteria have a cell wall, and we do not, the cell wall is, I would say, not even arguably, it is the most common choice for developing new antibiotics or modifying antibiotics to make them more effective, because we don't have one. So who cares what the antibiotic is doing, what kind of process it inhibits? We don't have any of those processes. We don't have anything similar to building a cell wall. So if you have antibiotics that can inhibit building a cell wall, you have antibiotics that will work to target the bacterium, but not target the host. You have the magic bullet. In chapter 7, when we talk more about antibiotics, we'll talk about antibiotics that do things like inhibit protein synthesis. We make proteins too. That makes it more challenging to inhibit bacterial versus human protein synthesis. So the cell wall tends to be the site for a lot of antibiotics. And while broadly the cell wall is made of peptidoglycan, gram-positive and gram-negative cells organize their cell walls in slightly different ways and have slightly different, well, sometimes significantly different modifications. So this section, when we start talking about the differences between gram-positive and gram-negative cell walls, this is also going to be one of those fundamentals, like if you don't understand this, like this is fundamental microbiology. I don't expect, if I go tag some of the ecologists, they might not know this part. But this is like fundamental microbiology. It will be very difficult for you to be successful in this class if this fundamental microbiology is a struggle. So that's why we start with this fundamental microbiology. So fundamental microbiology, bacteria that have a cell wall make it out of this compound called peptidoglycan. So peptidoglycan has, peptido, so proteins, Glycan, sugars, proteins and sugars. And the composition of the peptidoglycan in terms of the base subunits is consistent across bacteria. And I'll always say, as of our current knowledge, we have never discovered a bacterium that uses something that is not this. They all use peptidoglycan if they make a wall. So the carbohydrates, so there's two monosaccharides, N-acetylglucosamine and N-acetylmuramic acid. I'm not going to expect you to remember that. Even if you abbreviate them, that's great. NAG and NAM. N-acetylglucosamine, N-acetylmuramic acid. That's the glycan part. And so it's just a disaccharide. NAG, NAM, NAG, NAM, NAG, NAM, right? Like me, nagging, on and on, okay? So it just repeats. Then you have peptide cross bridges, and they are tetra peptides. 1, 2, 3, 4. Notice we don't even list the amino acids there. It is not terribly important. 4 amino acids, tetrapeptide. So that links them all together. And there you have peptidoglycan. Disaccharide, tetrapeptide, disaccharide, tetrapeptide, on and on and on. Okay. Now, fundamental differences of fundamental concepts in microbiology. Broadly, most bacteria that have a cell wall can be classified in one of two ways. Do you do gram stains today? In the morning? Okay, so afternoon people, gram stains. Okay. And it involves two things. One, how many membranes they have, and two, how thick their layer of peptidoglycan is. Those are two broad differences, but we'll talk about a few more. So a bacterium is classified as gram-positive. It has a single membrane, the plasma membrane that surrounds, all the cellular components. Then it has this very thick layer of peptidoglycan. One membrane, thick layer of peptidoglycan. Within that peptidoglycan, you'll find teichoic acids. They can help function in terms of things like cation binding. Okay, they're really good at that. And there will be two different kinds. You'll have some teichoic acids that are only associated with the peptidoglycan. Those are called wall teichoic acids. You'll have some that can extend down to the membrane. Those are lipoteichoic acids. I don't really care that much. Whatever, wall, lipid, I don't care. But I do care that only gram-positives have teichoic acids. I will ask you, list major differences between gram-positive and gram-negative. This is one of them. One membrane, one thick layer of peptidoglycan, teichoic acids. Somebody will always put purple. Purple? Because you can do a gram stain with other stains. It doesn't have to be purple. They're not purple. They're only purple if you do a gram stain the standard way. So 3 major differences. Fourth, they have a very thin periplasm. 4 major differences. So if you don't have to understand any of those words, but if you can spit them back out on the exam, that is what I'm looking for. You'll understand them later. I hope you understand them. But the beginning is memorizing that. You have one membrane, a very thick layer of peptidoglycan, teichoic acids, and a very thin periplasm. Okay, gram negatives. Well, let's think of all the things of gram positives. Well, gram positives, you highlighted gram positives only have one membrane. Well, so gram negatives have to be different. They have two. Gram positives have a really thick layer of peptidoglycan, so gram negatives have to be different. They have a thin one. We're going to skip teichoic acids for just a minute. Gram positives have a really thin periplasm. Gram negatives must have a bigger one. All of those things are true. So now, what about teichoic acids? Well, okay, gram-negatives don't have teichoic acids. That is true. And if you listed all of those things and you stopped there, you would get almost full credit on that question. Almost. Because you're missing the most important difference, I guess, in my opinion. And maybe for those of you going into healthcare or your patients who have a gram-negative infection. One of the most important differences between gram-positive and gram-negative is that in that second membrane, in that outer membrane, gram-negative bacteria have something called LPS, lipopolysaccharide. And I'm going to go to the next slide because it has a little bit more detail. So here we've got our gram-negative again. You can barely see this is the inner membrane, which is the classic like plasma membrane of the cell. You have this big periplasmic space, and in there is your really thin layer of peptidoglycan. Then you have this outer membrane that in some ways looks like another membrane, that's cool, but in other ways is very different. And this way that it's very different, the most important way is right here, lipopolysaccharide. So lipopolysaccharide has three components. Kind of embedded within the membrane is a molecule called lipid A. Lipid A is also what we call an endotoxin. It is an integral part of the cell. It's not something that it secretes. It's an integral part of the cell. But what happens is as the cell replicates or as a cell dies, it can release little pockets of LPS and therefore little pockets of lipid A, which overstimulate the immune system and can more rapidly lead to something like septic shock in a patient. So that lipid A broadly makes gram-negative infections very dangerous. Are they more dangerous than gram-positive? No, we don't deal in absolutes. But it is an inherent feature of all gram-negatives. They are inherently toxic because of that lipid A. Next, they'll have a core polysaccharide. This is one long molecule. The core polysaccharide is going to be a different polysaccharide for every species. Every species has their own particular core polysaccharide. And then they'll have this O polysaccharide, which is just another like long chain of polysaccharides. And that typically serves as the O antigen. So the O157, right, is that part of the LPS. So again, classically, I'll get a table like this. Gram-positive have thick peptidoglycan, have teichoic acid, they have thin periplasm, they have one membrane. Gram-negative have thin peptidoglycan, no teichoic acids, outer membrane, thick periplasm. This is great. You've got almost everything. But if you want full credit on this question, I want LPS. Oh, you could swap it out for 2. Have teichoic acid. Whatever. 2 or 5, I don't care. So again, this is like the fundamental microbiology. Like, truly, if I was able to ask you like one question, you have to answer the question correctly or you can't pass microbiology, this would be it. That's how fundamental it is to microbiology. So I'm telling you right now, this question will be on the next exam. It will also be on your final. And I say that. And in May, you'll be like, oh, what were the differences again? So keep reviewing those differences. And that is what allows us to do the gram stain. So those of you who've done the gram stain this morning, you understand these principles because you've actually done them now. Those of you who are doing the gram stain at 2 o'clock, have fun. It'll be a good time. I like doing gram stains now because I'm good at them. When I first learned, I was not that good. So this allows us to visualize, not like at the molecular level, but at least at the cellular level, make a call. Do we have the thick peptidoglycan and only one membrane? Do we have two membranes in that thin peptidoglycan, meaning I've got LPS and I've got a different kind of problem? That's what the gram stain does. So we talked about this when we talked about staining in chapter 3. So hopefully this is a little bit of a review for that. The first step is you use crystal violet. You could, in theory, use another stain, but classically, I forget his first name, but his name is literally Graham. It's his last name. That's why it's spelled G-R-A-M, because it was a guy, and that's why it's capital, because it's after a guy. It's his last name. So Graham used crystal violet, so we use crystal violet. We then use iodine as a mordant that binds to the crystal violet and makes this big crystal violet iodine complex. This is the step that probably, how many of you did it right the first time and you got it to look very perfect? Nicely done, very good. I did not, but Dr. Tal let us practice. So, after a few rounds of practice, I ended up doing okay. That decolorizing, now when you do it the Goldilocks, right? Not too long, not too short, just right. You rinse the crystal violet iodine complex out of your gram negatives because they have that thinner peptidoglycan. They won't hold on to it as long. But you don't do it so long that you actually wash it out of the gram positives, which have the thicker layer. So the alcohol dehydrates the peptidoglycan and should help keep it in there. But again, if you do it long enough, it will come out. I guarantee it. And sometimes it's like, I did it 10 seconds just like I did last time. Yeah, but you put on 2 extra drops and now it's toast. It can be very picky. So it's nothing you have done wrong. It's just a very picky stain. Now in theory, the gram negatives, right, should lose. They don't have very much peptidoglycan, so even if it dehydrates, big deal. Alcohol also denatures the membrane and allows that the crystal violet to come out of the peptidoglycan. So theoretically, and I believe this is a question that they often ask in the lab, what color is each step? Purple. Purple. Purple gram positive, clear gram negative. Purple gram positive, pink gram negative, because you use that counter stain. We love that question. At least I love that question. I know Dr. Tao did when I took it from her, because I remember having that question. But what about things that are different? So we do have stuff that's different. Not everything is gram positive or gram negative. And we even talked about this in the staining section, right? We talked about these cells that have mycolic acids that interfere with the ability to do a gram stain, because look at these things that are sticking out. This waxy substance. So they still have peptidoglycan. They have a cell wall. It's made of peptidoglycan. They have a single membrane, right? But they're not gram positive. Gram positive means you can do that gram stain and you just have the peptidoglycan and the membrane. Well, they have all this other stuff. And that layer of peptidoglycan doesn't look that thick either. But they're not gram negative because they don't have 2 membranes, and that looks a little bit thicker than a gram negative. They don't see a big periplasm. They're not gram positive, they're not gram negative. They're acid fast. So they're sort of like a gram positive because they have peptidoglycan and only 1 membrane, but you cannot do a gram stain on them and have it be successful. It just doesn't work. So instead, you would do an acid fast stain. Did you do that one today? Okay. Did it work pretty well? Yeah, that's a good one. They hadn't been doing it for a really long time, and I was like, no, let's try it. So a couple years ago, they were the guinea pigs. And then you have bacteria that don't have a cell wall. These are fun. Okay, and this one is also, I'm going to tell you. This is where people get tripped up. Mycobacterium is acid fast. It has mycolic acids. And I don't name these things, so don't be mad at me. Mycoplasma has no cell wall. I didn't name it. Okay. These are very, very different bacteria. So then what is that implication? Well, you can't stain them using anything that stains the cell wall. They don't have one. You can't treat them with any antibiotic that inhibits the cell wall. They don't have one. So Mycobacterium pneumoniae is usually what causes what people call walking or atypical pneumonia. People always think pneumonia is like strep. So here's your penicillin derivative. And then, right? Okay, so mycoplasma, no cell wall. Mycobacterium, acid fast. And then you have archaea, which again, either don't have a wall or they have a different wall than peptidoglycan. And really the difference is pretty subtle. They do use N-acetylglucosamine. They do make the tetrapeptides and use the cross-linking. They don't use the NAM. They use this one instead. So they use a different carbohydrate. So one different carbohydrate. And instead of the D form of amino acids, which is what peptidoglycan uses, archaea use the L form. And that has a couple of implications. Antibiotics like vancomycin work only against the D form. Lysozyme, which is a component of our tears, can only break apart the junction of N-acetylglucosamine and N-acetylmuramic acid. It cannot break apart the cell wall of archaea. So it's good that we don't know of any archaea that cause infection, because our own basic defenses wouldn't be very good against it, and most of our antibiotics wouldn't work either, because their cell wall is so subtly different that it actually makes that big of a difference. We'll pick up on Thursday talking about more damage to the cell wall, and then we'll keep moving inside of the cells.